Hi, my name is Paris Martin and I'm currently completing my third year of a Bachelor of Science at JCU, majoring in marine biology. I recently completed a 6-week placement with CSIRO in Perth, Western Australia. With a strong interest in environmental DNA, I was placed with Dr. Joseph Perkins, whose postdoctoral research focuses on harmful algal species in Western Australian estuaries using eDNA. My role involved assisting with field sampling, including water collection and filtration for eDNA analysis, recording environmental metadata, preparing filtration equipment, handling preserved samples, and participating in DNA extractions. Throughout my marine biology degree, I've always enjoyed fieldwork and conservation, but I wanted to expand my practical skills in molecular biology. As eDNA is becoming an increasingly important tool in conservation and environmental monitoring, this placement aligned perfectly with the skills I hope to develop during my degree. My primary technical goal was to develop skills in estuarine field sampling. Unfortunately, due to poor weather, we were unable to complete the planned estuarine field work. Although this was initially disappointing, it led to one of the highlights of my placement. We instead travelled to Rottnest Island to conduct marine field sampling. Although there wasn't enough time to complete microscopy or GIS work, I was still able to experience the complete eDNA workflow from field sampling through to lab analysis and data interpretation. My communication skills also developed significantly through the placement. At the beginning, I was quite hesitant to speak up because I was still learning and didn't want to ask the wrong questions. However, as I became more familiar with the project, I found myself regularly discussing my progress with Joe and the research team. I also had the opportunity to prepare fieldwork protocols and at the end of my placement, present my quick comp trial results to the team. Looking back, this was something I never would have imagined myself doing on the first day, but by the end of the placement, I felt much more confident communicating scientific concepts and discussing my work. The placement also helped me develop greater self-awareness. At the beginning, I felt nervous about working in a professional research environment because I had limited lab experience and often doubted my own abilities. Looking back now, this is probably where I experienced the greatest growth. The team created such a supportive environment that I quickly became comfortable asking questions, discussing what I was learning, and talking openly about the areas I found challenging. By the end of the placement, I felt far more confident in both my technical abilities and my ability to contribute to a research team. My self-regulation skills also improved considerably. Balancing university with other commitments has always been something I've found challenging, so completing this placement while continuing 2 university subjects required me to become much more organised. Throughout the 6 weeks, I learned how to manage my workload, prioritise tasks, and meet deadlines while maintaining a high standard of work. Keeping organised records and receiving positive feedback from Joe about both the quality and organisation of my work gave me much more confidence that I was developing the professional skills needed in a research environment. Finally, my teamwork and interpersonal skills developed more than I expected. Before the placement, I generally preferred working independently, but spending 6 weeks alongside such a supportive research team completely changed my perspective. Whether we were out in the field at Rottnest Island or working together in the lab, I really enjoyed collaborating with people who were passionate about marine science. Looking back on the placement, I believe I achieved all of the goals I set at the beginning. Although my technical objectives changed because the planned estuarine field work couldn't go ahead, those challenges were replaced with equally valuable opportunities. The greatest growth, however, wasn't technical, it was personal. Building confidence in my own abilities and realising that I could contribute meaningfully to a professional research team has been the most valuable outcome of this placement. I developed practical skills in harmful algal bloom research, including field sampling, DNA extraction, cubic quantification, PCR, gel electrophoresis, and analysing data in R. One of the most rewarding parts of the placement was seeing our samples progress through each stage of the workflow and understanding how every step contributes to producing meaningful scientific outcomes. Working alongside such a diverse team of researchers also gave me valuable insight into the many different career pathways within marine science. Through conversations with the team, I gained a much better understanding of research careers and the importance of building professional networks. I was incredibly grateful to be invited back for another studentship in December and to attend the Australian Marine Science Association conference in Canberra with the team in September. Being offered these opportunities gave me confidence that I could continue contributing to this field and reinforced that I was on the right career path. Perhaps the biggest impact that this placement had was changing the direction I thought my career would take. Before starting at CSIRO, I expected to move primarily into field-based marine science after graduating. However, this experience showed me how exciting molecular research can be and sparked a genuine interest in pursuing an honours project focused on environmental DNA. Seeing how eDNA can answer such a wide range of ecological questions and the growing role it's playing in environmental monitoring and conservation made me realise this is an area I want to continue exploring. When I return to Townsville, I hope to build on this experience by volunteering wherever possible and continuing to develop my skills in environmental DNA and harmful algal bloom research. I now feel much more confident about pursuing a career that combines both fieldwork and molecular research.